If I may, I'm going to take 1 minute to first thank the organization that invited me to be at Kernel. It's a double pleasure. First, because it's nice for an animal breeder to be invited by plant breeders. That indicates that as a minimum they read your papers. And secondly, because he gave me an opportunity to return to Kernel, where I spent a year working with Professor Charles Henderson. He was in animal sciences, and he was the developer of mixed model methodology. Actually, he started this in his PhD thesis in 1948. And while at Kernel, I also took courses with Charles Searle. He was a statistician originally from New Zealand in the biometrics unit, and also with Dale Van Vleck, that he retired from Kernel and moved with the USDA at the University of Nebraska-Lincoln. First, we talk about complex traits. Why are traits complex? Well, the first thing is because if you have any friend in biochemistry, they have these charts in their walls. And when we talk about a phenotype, this is the result of an exquisitely integrated set of reactions that are working in a concerted manner with the environment. It also must be true, mechanistically, that epistasis is pervasive. And a simple reason is this one. If you consider any simple biochemical cycle, such as the Krebs cycle, there are several enzymes that work in concertation. And we have this paradigm, one gene, one enzyme. So any simple pathway has to be affected by several enzymes working together. And the enzyme production and output is regulated by a nonlinear logic called Michaelis-Menten. And it is, of course, extraordinarily naive to express an output from this system as just the sum of 12 things are adding up. Actually, if you kill one of those, the whole cycle stops. So that is indication that interaction is a fundamental force in biology. And we do have evidence that these interactions exist using molecular data. Now we have started to understand a little bit of the architecture of interactions, at least in Drosophila melanogaster. This is where it comes from Trudy McKay's laboratory in North Carolina State, where they have constructed simple epistatic networks, pair-pair interactions between several markers in two populations of flies for two traits, starvation resistance and chill coma recovery. The second one believed to be very strongly epistatic. Now, as you may know, at least those that work in applied animal breeding, there is a central tenet of quantitative genetics that postulate that by adding things that are a linear combination of some effects at some loci, let's say k number of loci, indexed by a random variable that indicates the genotype of that locus, and that each of these random variables may have a distribution, which could be, for example, the Hardy-Weinberg distribution, but jointly the W's may be defined by a linked disequilibrium distribution. If you add this up, you produce something that's called the additive genotypic value. And today, in the era of markers, the idea is to fill this bucket called the additive genetic value using markers. And this white part is the so-called missing additive genotypic value. And of course, the more markers, the more relevant these markers they are, eventually we'll be able to fill the bucket, assuming that the bucket exists. It is the theory that postulates the bucket. Perhaps it does not exist. Now, an important matter that actually most quantitative geneticists view as a second-order problem is that of epistasis. It's such a difficult complex that you have to engage all these superheroes to help you. Why is it so? Well, it's because we do not have a good theoretical body for dealing with epistasis, at least from a variance-covariance component decomposition. The available theory that dates back to Clark Cockerham in North Carolina State and Oscar Kempthorne in Iowa State says that given certain conditions, you can decompose the genetic variance into an additive, dominance, and epistatic components. And these epistatic components can be further decomposed into additive by dominance, additive by additive, dominance by additive, dominance by dominance, etc., 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 etc. But the theory holds if there is no selection, no inbreeding, no assortative mating, no mutation, no migration, linkage equilibrium, and no linkage. And if you believe in this assumption, then you believe in the tooth fairy. Okay? So all assumptions are violated. Sorry, folks, flush the theory. Now, the good news is that many of my colleagues in quantitative genetics say, well, you don't need to worry about epistatic. because then most of the variance actually translates into genetic variance. And there is this influential paper by Bill Hill from Edinburgh where they studied several systems, some known to be highly epistatic, and most of the variance, 50 to 100% of the variance, turns to be additive. And this makes people happy. But actually, they are committing kamikaze, suicide, because of the following. This implies that this places an upper limit on how much you can learn about this so-called genetic architecture using statistical methods. Because everything turns out to be additive irrespective of whether they are or they aren't. Okay? So, paraphrasing somebody will cite in a few minutes, the additive model denies what it is and explains what it's not. It's a splendid metaphor, but consider this metaphor. Okay? The doctor is giving good news to a patient that has a knife in the back, and they say, oh, you are okay. All right, and so this that explains what it is and denies what it is, and was actually inspired by Rousseau. He has exactly this in his book, La Nouvelle Héloïse, so you probably know who Rousseau was. Now, in quantitative genetics, we deal with two main tasks, inference and prediction. And quite often, these tasks cannot be distinguished from each other, but it's important to keep in mind the differences between inferential and predictive tasks, because the tools and the ways of reasoning are entirely different. In inference, we start with the following. We have data, pedigree, genomic data, phenotypes, etc. Then we invent a model. That is, the model may be suggested by mechanistic considerations or because you fell in love with la nouvelle vague de la statistique, and then you have some parameters that you have, that you have to learn, fit. You estimate the parameters, and the question is how do you estimate parameters? Well, you choose estimators. So you talk to your favorite statistician and you'll get a recipe. Use moments, use likelihood, your Bayesian, and then the statistician will try to convince you, based on theoretical arguments, that the estimates are close to a state of nature. Of course, a state of nature you can never observe. And more recently, they talk about the fundamental oracle properties of a good statistical method. That has to be either that you have to hit the true model, and if you hit the true model, then the value of the parameter is learned asymptotically, namely when you have an infinite amount of information. Okay? So this is a very optimistic view. First, you never know the state of nature, and then by accident you have to hit the true model, when it may be that a true model does not exist, and then learn the parameters of the model by having an infinite sample size. Now, in today's world of genomics, I think you'll agree with me, we have very small samples relative to the number of things that we want to learn from the data. And irrespective of whether you're a likelihood-based or a Bayesian-based person, you have to recognize that whenever you have more parameters than data points, the unobservables are not identifying the likelihood. The likelihood can answer at most n questions. And if the n questions are answered, then you are saturating the model right away. So the Bayesians, of course, have a solution for everything, and they bring this external information represented by the prior, and they have invented no less than 20 different prediction machines that goes to base A to base zeta delta. And it's useful to remember a voice from the grave. Oscar Kempthorne, one of the most ferocious anti-Bayesian person I've ever met in my life. And he said, reporting only Bayesian estimates, each based on the prior of the person who obtained them, will butcher the process of science. And you may be surprised, if you know a little bit of me, that I was partially responsible for introducing Bayesian methods in animal breeding and MCMC, that will be quoting this statement for Kempthorne. And the reason is that we Bayesians had a counter-argument when the number of parameters was smaller than sample size, because we could argue asymptotically we'll be able to learn. But that's not happening. Actually, the number of unknowns is growing faster than the number of parameters, because when we finish genotyping with the last model of the last chip, a new chip comes up. And when you have genotype everybody with the last chip, they tell you, no, you have to do sequences, and so on and on and on and on, so P grows faster than N. Okay, prediction, on the other hand, after having established that inference postulates the existence of these parameters that you can never observe, so at best you can say that given that the model is true, you are going to get close to the parameters, but you'll never be able to observe them. On the other hand, you have what is called the predictive inference approach. And let me quote a fragment of the book by Geiser, a statistician, late statistician of the University of Minnesota. He said, clearly, hypothesis testing and estimation, as stressed in almost all statistics books, involve parameters. This presumes the truth of the model and imparts an inappropriate existential meaning to an index or a parameter. Inferring about observables is more pertinent since they can occur and be validated to a degree that is not possible for parameters. So then we move into constructing what are called predictive distributions. And the Bayesians, given a certain model, they can develop a predictive distribution by integrating the way that you think the future observations will arise by taking into account the uncertainty about the parameters of that model. This is called a posterior density. This is the distribution of future data given the known parameters, and this act of integration will eliminate the uncertainty in a probabilistic manner about the parameters that you cannot observe. And then you develop a predictive distribution. But what happens? That quite often you are not sure about the model. You may have several competing models. So the Bayesians have another answer, is that, well, if you do not know the model, you have to take the model uncertainty into account. So then you take this distribution and integrate it over the posterior distribution of the model. So a model, let's say you have a finite number of models, then each model has a prior probability, typically the same for every model, but then a posteriori will have different plausibilities, so you use the posterior probabilities to take into account the relative uncertainty about the model. So I use what is called Bayesian model averaging, and one of the continuous variants of Bayesian model averaging can be implemented using an algorithm that's called reversible jump Markov chain Monte Carlo. That is notoriously difficult to tune, and of course, it's totally unfeasible in multiple dimensions. You'll probably die before it converges. Now, so the end of the story is that we deal with complex systems. We have to accept, as I often say, that you have to surrender in the age of complexity. Does that mean that you should not do anything about it? Well, of course not. I was talking with the graduate students prior to coming here about how successful breeding has been, dog breeding. The dog breeders have not used genomics, have not used pedigree selection. They just, they select the dogs according to where they want them to bark, to sleep, to hunt or to do, and then they do consanguinity. There's not much science, and yet, if you have seen a Chihuahua and a Great Dane, you see that they have been successful. So that's breeding without understanding. Now, so from now on, I'm going to talk about prediction machines. And I suppose that some of you have heard about best linear bias prediction using pedigrees. That's something that, as I said, was developed by Charles Henderson here at Cornell. And in the 70s, Cornell was really the center of action in terms of animal breeding and applications of this paradigm to dairy cattle genetic evaluation. Then came genomic blob, in which is essentially blob with a pedigree matrix replaced by a genome-based relatedness matrix. Some of you may have heard about support vector machines in regression or classification. This is part of the Vapnik statistical learning paradigm, and they are considered to be the state of the art in many problems from a non-parametric perspective. I'm sure that you, being mostly plant scientists, are aware of spatial problems, and kriging was a technique that was developed in geostatistics in South Africa for mining. And some of you may also heard about kernel machines in computer science. And all these are special cases of something that has a very scary name, reproducing kernel Hilbert spaces methodology. And it's indeed scary. This gentleman here is David Hilbert, one of the most influential mathematicians of the modern times. He was at the University of Göttingen in Germany, and you have heard about Hilbert spaces, some of you, a very rich space of functions. I have no idea whether the Republic of Congo made a stamp in honor of Hilbert, but there he is. So he was obviously important, at least to the Congo. And the basic idea here is, if you forget this part, which I'll try to explain in a minute, this is like a sum of squares, okay? And so here we have a parametric part. These betas are what statisticians call the fixed effects. The U's are what called the infinitesimal genetic effects. And then enter into the picture a battery of inputs that could be markers. DNA markers, but could be proteins, could be a string, could be a graph, could be an image, could be anything. And then we say, well, I do not know really how these inputs relate to the phenotype, so I'm going to assume that there is a function, g of x, that lives inside of a Hilbert space, and I'm going to minimize this objective function, which is the sums of squares of the residuals, plus a term, which is what statisticians often call a penalty. And the penalty is the squared of a norm in the Hilbert space. So this is what is called a functional. So as opposed to a standard calculus problem in which you try to find the maximum or the minimum of a function, you find the point, here we're trying to find a function that minimizes this functional in a Hilbert space. And this is a horrendous mathematical problem. It's called variational problem in physics. And the solution was found by my colleague at the University of Wisconsin, Grace Waba, when she was a graduate student. Grace is a member of the National Academy of Sciences in the section of Applied Mathematics and Statistics. And they found that the G function of whatever, inputs, pedigrees, proteins, DNA markers, shoes, it doesn't matter, is actually a linear function which looks very much like a regression coefficient, where you have alpha j regression coefficients, one for each individual in the sample, plus an intercept, times something that is called reproducing kernel. And the reproducing kernel is just a transformation of your input data, which may be linear or nonlinear depending on the problem. Okay? There is no general rule to guide you on how you should construct that kernel, but we will examine some in a minute. What you should note is that if we have n data points, there are n plus 1 coefficients, so it means that you should shrink something a little bit, otherwise it will not fit in the room. Okay? If you have room for n and you try to fit 1 million, with 1 million be larger than n, then you have to do something like that film from the Marx Brothers, A Night in the Opera. You know, they were trying to put 30 people inside of a room in a boat. So you have to start chopping heads, cutting feet, cutting arms, and that's exactly what shrinkage does. Okay? So accepting that the solution to this problem is a linear function on reproducing kernels, and they're called reproducing because they have certain algebraic properties. Under the integral signs, there are some reproducibility features. Then you can insert this entirely non-parametric structure into the logic of the mixed model and have a semi-parametric model. And the theory, by making an analogy with a certain class of Bayesian model, dictates that this unknown vector of alpha coefficients should be viewed as drawn as random from a normal distribution with variance sigma square alpha pre-multiplied by the reciprocal of the matrix of kernels. So the matrix K of order n by n will define a matrix of similarities among n individuals in the sample. And these individuals do not have to be necessarily genetically related. All it suffices is that you can construct notions of distance among individuals, for example, based on molecular similarity and not necessarily on lines of descent. And you can, of course, construct kernels in many different ways. So I'll show you some of the most popular kernels. There are two things that enter into a kernel. First, one is the notions of distance between individuals. The most widely used one is the Euclidean distance. So for example, if I ask what is the distance between me and everybody here, I genotype this gentleman here that happens to be a Wisconsin graduate. I genotype me, and then I look at how similar we are to each other. And then I use a bandwidth parameter to either exacerbate this distance or to attenuate it. And the way you choose that bandwidth parameter is either by cross-validation or by estimating it directly from training data, which is a horrendous problem by itself. Mathematicians criticize the Gaussian kernel because they say they are better kernels and they kill each other on kernels, but practically speaking, we have found no instances in which fancier kernels are better than the Gaussian. So for all practical purposes, this seems to work well. So if you use a Gaussian kernel, essentially you transform these inputs into a nonlinear manner. If you use pedigrees, these Aij suppose are the entries of a kinship matrix, there is nothing that will preclude you also to transform it nonlinearly and also to introduce some bandwidth parameter to either attenuate or exacerbate distances between pairs of individuals. And a kernel that I introduced here, which is thicker tail, I'm sorry, I forgot to turn off the volume of this. I hope it doesn't bother you too much. Is what is called the simile of a multivariate T-kernel. It behaves very much like a Gaussian kernel, except it has thicker tails, and the bandwidth parameter are the degrees of freedom of that T distribution, which is a positive parameter. It goes between 0 and infinity, and you can also either estimate or learn it from cross-validation. In our experience with kernel-based method from a genome-enabled prediction perspective, we have found that the best approach to prediction using kernels is to use what is called a multi-kernel fitting. As opposed to estimate the bandwidth parameter, suppose that we settle on the Euclidean distance, and suppose that you settle on the Gaussian kernel, then the next question is, how should I tune the bandwidth parameter? Well, one way around it is to consider several kernels simultaneously and fit them all together, okay? And then assess the predictive ability of that multi-kernel fitting with cross-validation. And quite often we have found out that using several kernels together leads to better predictive performance than using a single kernel. And there is nothing at all in theory that precludes abandoning the kinship matrix derived from pedigree, because kinship matrices from pedigrees and kinship matrices from markers are not necessarily representative of the same process. The kinship matrix from a pedigree is the expected value of genetic relatedness under some assumptions, like no selection, no mutation, no migration, no whatever. Whereas the genomic relation matrix assessed from markers is a realization of a process that is not necessarily the same stochastic process on which we base our assumptions when we construct a kinship matrix, okay? So they too may be telling you different stories, and they too may complement each other, predictively speaking. So then the problem reduces to one in which you have several random terms, the infinitesimal effect and 4 variance components associated with each of the kernels, each of which is indexed by a specific bandwidth parameter. And sorry, I forgot to say, the basic idea of the different bandwidth is to create either global or sharp degrees of similarity. For example, if I decide that I don't want to be too similar to him, molecularly, I use a very sharp kernel that will allow my genotype to be compared only with the genotypes of individuals that are very proximal to me in state, okay? On the other hand, I may choose that I would say, let's say, a more egalitarian kernel in which I will give equal opportunity to all of you, no matter whether you are red, green, yellow, blue, or whatever, to be similar to me. And then I would choose a kernel that is more gentle, and it will create similarities even among individuals that are much more dissimilar than they should be. Now, some questions ask, why is it that these guys can capture epistasis even when you construct kernels that encode only additive information? For example, when we use SNPs, we normally code these SNPs are either -1, 0, 1 for little a, large a, little a, large a, large a, and actually I'm using only additive encoding information. So why can it encode epistasis? Well, the answer is simple. I'm using pictures, so I could not use the animations and sequences, so bear with me in the first part of the slide. Suppose that this simple regression model describes a two-locus model in which beta 1 and beta 2 are the additive effects of each of the two loci, and beta 1, 2 represent a joint effect, namely epistasis. If you assume that the x's are continuous, which they are not, and consider the first differential, the only differential with respect to x1, that is like the impact of an allelic substitution. What happens when we go from 0 to 1 and from 1 to 2? And then if this derivative depends on what's going on in locus 2, then we say there is epistasis, right? That means that whatever you observe at the finite peak level depends on an interaction between the two loci. Now, if I replace this simple model with a kernel-based model, specifically a Gaussian kernel, and I take the derivative with respect to the vector of genotype or with respect to a single SNP, let's say xj, then I find out that whatever happens when I replace an allele at SNP j depends on the genotypes at all the other loci. So these kernel methods are able to capture forms of interaction that are not contemplated by statistical interaction. Statistical interaction always based on a linear concept. The model, the derivative of the model with respect to the parameters is linear with respect to the parameters. This is here is linear with respect to the parameter, but non-linear with respect to the inputs, which is exactly what's happening here. The second question, this is, you probably most of you know Bill Hill from Edinburgh. He always asks me the same question. Oh yeah, all this is very nice, but I cannot get a breeding value. Well, unfortunately, fortunately rather, you can. And the way you deal with that is just by kernel construction. You may construct this part, which is parametric. This is the marked infinitesimal, this is the infinitesimal value. This is the additional value that you could mark with markers. And then you have several nonlinear Gaussian kernels that may capture whatever epistasis you can capture, if you can capture it. So then it is not true that these methods cannot produce a breeding value. They can, and more. A third question, and some of my colleagues in animal breeding have really gotten carried away, is they say, well, I can construct a genomic relationship matrix for additive effect. In each SNP, I have two variables. I can code, there are three genotypes, so I can have two dummy variables, one for additive effects, one for dominance. Why not constructing a genomic relation matrix due to dominance in the same way that I can construct one due to additive effects? And if that is the case, I can fit these two things together in a variance component model and even construct another kernel by doing the element by element multiplication of the entries of these two matrices and capture additive by dominance marked epistatic variance. Why not? Well, my younger colleagues should remember Cockerham and Kempthorne that unless this kernel, unless everything is orthogonal, namely unless the kernels are orthogonal, then there is some sort of a collinearity between the dominance kernel and the additive kernel, simply because in the absence of inbreeding, if you think in terms of pedigrees, you can construct the dominance relationship matrix from the additive relation matrix. So there is a built-in collinearity. So in order to learn additive and dominance variance separately, especially from large and structural balanced data, you have to construct orthogonal kernels, and that's not a single matter. So do not take seriously claims about dominance and epistatic variance coming from animal breeding because they are based on naively constructed genomic relationship matrix. And we are far from being able to say this is the way that you should construct genomic relation matrices. Now, going back to the idea of prediction, to learn about observables, you have at some point to make an honest test. I have a prediction, I have a realization, therefore I can say I have predicted well or bad. So the way we do this is we have data. These data are generated according to some unknown process, so you invent a model, okay? The model of your choice, based on structural biology considerations, or just based on a computer scientist that knows beans about biology, but will be able to construct a good model because these guys do know how to deal with complex systems. So then you split the data into training and testing things, And the fitting process describes how well the data describes the training set, but then you want to make statements about yet to be observed data. When you eventually observe the data, you know that you have observed the prediction error, okay? You predict the phenotype, you have the prediction. If you are lucky, you predict exactly. If you are unlucky, you predict awfully. But you can't construct the prediction error. Now, if you do this only once, that's like doing an experiment with a sample size n equal to 1. During lunch, we were talking about these guys that find the tooth of a wild boar and they make stories about what happened 100 million years ago. I still don't believe it, in spite of the arguments that were advanced to me at lunch by brilliant graduate students. So then you have to do this several times, and based on that several times, then you construct an uncertainty distribution, which sometimes you can, if you can replicate the cross-validation, but sometimes you can't. Because, for example, if your training set is generation t and the testing set is generation t plus 1, that happens only once. How do you replicate something that happens only once? Well, you pseudo-replicate. You use some sort of clever bootstrapping mechanism, and then you can construct an approximation to the cross-validation distribution. So this is roughly a very rapid overview, perhaps not too rapid, of prediction with kernel methods, and also try to distinguish the difference between inference and prediction. I'm here interested in prediction. I'm not interested in learning about the state of nature, because I don't think that state of nature can be learned for complex traits, especially with any smaller than p. Now, some excitement from plant breeding. Typically, the excitement often comes from simulations, okay? And as I said this morning, jocularly, but to some extent seriously, quite often the simulation studies reported tend to reflect more the state of mind of the authors than the state of nature, okay? You know what I mean. You simulate until you find what you wanted. So there is this paper that came recently in the journal called G3. It came from Iowa State, where they simulated two situations, one in which there were 10 chromosomes with 2 QTLs, 2,000 markers per chromosome, another one that was completely epistatic. It was, there were only additive by additive epistasis within each of the 10 chromosomes. And then they compare these two situations at different levels of heritability, 30% and 70%. Animal breeders, when they see a heritability of 70%, say, wow, these plant breeders are really lucky. Well, there is an important difference. As what plant breeders call heritability is actually the heritability of a mean in the sense of Faulkner, because they average over blocks or replicas, etc. So they normally have very stable observations. And they compare a bunch of methods. And in the top, we have heritability 30%. In the bottom, heritability of 70%. The only thing I want to bring to your attention is here we have a suite of parametric models. And here we have reproducing kernel Hilbert spacer regression. This is an additive model. Okay? The kernel is a non-parametric method. We have complete additivity, no assumptions made. Roughly, we made predictions that are quite similar. Now we move to 70% heritability. Here are the parametric models. All they use is beautiful additive theory, blah, 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 blah. And here you have the reproducing kernel Hilbert spacer regression. It's slightly worse, but nothing to go about the war. You have to remember, maybe you cannot see, is these are predictive correlations. And what really matters is the square of the correlation. So if the difference correlation is 0.03, that means that the difference in the square of the correlation is 9 times 10 to the minus 4, something like that, okay? Nothing to go to war about it, except scientists. Now, when we move to this stylized form of epistasis, then we see clear methods in favor of the non-parametric methods. And here you have reproducing kernel Hilbert spaces. Nadaraja Watson is a widely used estimator in econometrics and support vector machines, but not big differences here. But there is a big difference when you move to heritability 70%. So that suggests, to the extent that the simulations in some sense reflect the state of nature, which of course we know that's not the case, that the kernel method do better. This is a paper by Paulino Perez Jose Crosa from Mexico, dealing with wheat, also published in G3, where they compare several parametric prediction machines, the Bayesian lasso, Bayesian regression, base A, base B, against several non-parametric methods, and the underlying are the cases in which a particular method was better than the others. And here you observe something to which I will return later, that if you average out all these comparisons, base B, which is in some sort of Bayesian pseudo-variable selection method, did not win any tournament, but on average, smelled like a flower. You know, are those guys that never win an election, but they end the life in the unpolluted states of never being touched by, by politics. Okay, something that the Bayesians and the non-Bayesians like to say in the predictive world is they say, well, if you have several models and you are unsure about how plausible these models are, in theory, on average, you predict better if you average this model in some sense. And we tested this with pig data. It was a paper that came published in a journal of animal breeding and genetics. And what we did is we compared several RKHS implementation. One with a local kernel, two with an intermediate kernel, three with a fat kernel, a global, some sort of a social democrat kernel, so to speak. Then we fitted models with kernels 1 and 2, 1 and 3, 2 and 3, 1, 2, 3. And then we did two different ways of model averaging, one in which we just average the predictions, just using an arithmetic average, and another one in which we used the Bayesian logic. We computed the posterior probability of the model and averaged with respect to the distribution, assuming that the models were equiprobable. Okay? So then the model plausibility is dictated by something called the Bayes factor. At the end of the story, we found something like this. No differences could be found among machines, except that the first kernel, the very local one, tended to predict slightly worse than the other ones. And Bayesian model averaging, no matter how we did it, 8, 8 star, or 8 double star, was not better than using 3 kernels separately. Okay? So that's good news, because that means that you don't have to go through the trouble of using Bayesian model averaging that is more computationally taxing. I think you know this gentleman, Nicolas's law. He was here. And I like that paper. I actually have used this table many times because he and co-workers compared several methods over 18 trials. Okay? The barley, wheat, maize, etc. And I want to bring to your attention the predictive correlations average over trials. These are based on cross-validation. And if you see the different methods, 0.56, 56, 54, 56, 56, 54, 59, nothing to go to war, but the Hilbert method came slightly better. There were some strange things going on, because if you look at the training set correlations, there was some indication that the Hilbert space model was overfitting, but yet predicted the best. That's a bit unusual, but that's what the paper reported. So if you leave things as they are, you conclude, okay, there are no serious difference among prediction machines. And this is what we really observe in animal breeding, that seldom a prediction machine is uniformly blessed. Now, really, okay, how many of you like malt whiskey? I like malt whiskey, but I have no idea. You give me Glenfiddich, which is a cheap malt whiskey, fine. You give me this guy, it's much better, fine. You give me Lagavulin, which is an islay, it's even better. So in order to classify whiskeys, you have to learn how to classify whiskeys. So this is a whiskey ranking that I picked up about a year and a half ago. So the top of the ranking was Daldane 14-year-old's connoisseur choice from Scotland, still in the United Kingdom. Okay, what do the whiskey secrets tell us? Well, they have to replicate comparisons, but never get drunk, okay? And the second one is to use pair comparison, something you learn in Statistics 100. If you have some rationale for comparing things, like with a block design, the variance of the difference is smaller that the variance of the difference if there is no pairing, because the pairing induces a correlation, and the correlation is positive, so the variance is smaller. So if you take Hess law data, this is my post-publication analysis of Hess law. Too bad I didn't do this. We have 18 points in which we can compare random regression blob, it's called G blob, actually genomic blob, against Hilbert. And in 18 of the comparisons, even though on average they were very similar, 16 out of 2 was a victory for Hilbert. Now, if you then compare random regression, Bayes C pi, which is an Iowa State construct based on the idea that you can do variable selection meaningfully, against Hilbert, again, you observe that Hilbert defeated Bayes C pi 16 out of 18 times. So if you think that Germany's 7-1 against Brazil was a decisive victory in the recent World Cup, you should agree that it's an even more so decisive victory. It's like playing Tahiti, okay? So if this doesn't convince you, under the null hypothesis of the probability of victory is the same, you can reject the null hypothesis at 1 per 1,000 level, and if you construct a confidence interval for the probability of victory, there you have. So the decisiveness of the victory of reproducing kernel over Bayes C pi, and the same would apply to genomic blob, is beyond reasonable doubt. Now, what can we do further? First is that training a prediction machine does not end up in doing it only once. You can actually use bootstrapping using an old idea by Bryman from the University of California, Berkeley, in which you can imagine that you have an infinite sample size of training set, but instead, and the way you do it, and you emulate it by doing bootstrapping from the sample. And then for each of such bootstrap samples, you contract the prediction, and then at the end, if you take the average, you have with a prediction that has exactly the same bias or lack of bias properties as the original predictor, but with a variance that at most b times smaller. Okay, and I have verified this conjecture. Here's a paper was published in PLOS ONE. This is simulated data, but I found the same thing with this famous WIT data set. And in 1,300 comparisons, we found that the bagged genomic blob, namely resampled genomic blob, was better than G blob, that it produces less overfitting, it predicted better, and produced smaller mean square error predictions. And the superiority comes primarily from situations in which you do the regularization of the prediction machine incorrectly. If you do it properly, there are no clear advantages of bagging. Now, if you can emulate an infinite supply of training sets, then you can solve an unsolved problem in animal breeding or in plant breeding. And you want to have not only a single measure of cross-validation reliability, but a candidate-specific measure. So you can emulate the concept of prediction error variances, and you do it. This is what you expect from the model, and this is what you get empirically. And there is no relationship whatsoever. Why? Because the theoretical prediction error variance that comes from the methodology does not look at the phenotypes. It just looks at the data structure of the various component decomposition. Whereas the one that comes from the repeated bootstrapping looks at predictive performance, how close you are to your true target, which is the phenotypes in the testing set. You can construct not only additive kernels, but also dominance kernels. You can measure the relative contribution of the additive dominance and additive bidominance if you get adventurous. Of course, you have to be cognizant of the problems of the lack of orthogonality, so you have to enhance. You can assign different bandwidth, for example, to markers in Hardy-Weinberg equilibrium, markers that are not in Hardy-Weinberg equilibrium, if you choose to retain them, differential ways to differential level of heterozygosity or whatever. Something that is coming up in human medicine, you may know or not know that so far whole genome prediction in humans does not have any clinical value whatsoever. For bladder cancer, it does not add anything at all over knowing if the guy smoked or did not smoke. In melanoma, it enhances the area under the curve marginally. There are very few documented cases in whole genome prediction being something that clinicians can use to advantage. There is a group from the University of Chicago led by Nancy Cox where she used what is called polyomic prediction. From DNA to phenotypes, there is a lot of stuff going on in terms of protein output, metabolic information. Should we use intermediate layers and found that by several diseases, then we could start talking about using omics for predicting human disease events. And something we should not neglect, especially with animals and plants, and of course humans, is that the environment is really powerful. So instead of spending so much time in genomics, proteomics, metabolomics, and ergonomics, or whatever omics, we should spend more time in measuring the environment. And I think this paper is paradigmatic of how genome-enabled prediction can be enhanced by taking environment into account, modeling more properly, and then building up some sort of a measure of G by E. So what matters is the statement that by taking into account G by E, predictive accuracy was enhanced by 20%. That's not a minor enhancement. I'm getting to the end. Carl?
Yeah.
Am I okay time-wise? Okay, 2 more minutes, and this will take 2 more minutes. This P to N ratio is a problem and will become even more so, okay? So, the gurus say that this will solve all problems because the causal mutations are there. Well, first thing that we ought to discuss is what is the meaning of causal mutation? What is cause? Are the genes a cause or an effect? Well, that's an arguable matter because in cancer, for example, we all carry oncogenes, and we get cancer if the tumor repressor genes fail to act in some cancers. And why do they fail to act? Well, because there is an environmental force that screws up the tumor repressor gene. So what is the cause? Is it the gene or is it the environment? Well, once you agree, okay, now we have the terminology, I know what you mean. Then the gurus and companies think that having a sequence of bases all over the genome is going to tell you the story. It's like they say, okay, well, what's the name of that highway? Do you remember that film in the 60s called Highway 66? Now, these guys that got into a Mustang and visited everywhere in the United States. I mean, if you look at the map, you have no idea what the United States is, but it will tell you Wichita, whatever comes after, Wyoming, Laramie, the works, the Mount Rushmore, but will not tell you anything at all about how people function, how they live, how they interact with each other. So it's a very naive view, in my opinion. Now, some of my friends, many of whom I am personally responsible for convincing in becoming Bayesians, they continue inventing Bayesian prediction machines. The first thing that we need to accept is when P gets much larger than N, the methodology may collapse computationally. Not because of time, because in today's world of parallel computing, you can parallelize your Markov chain Monte Carlo and run short chains in 30,000 computers simultaneously, say, for secular. So it's feasible. But inferentially, what will happen is the more markers and the more DNA information you put into the machinery, that's happening with sequence, the coefficients are necessarily shrunken by the priors, because this is the story of the Marx brother. If you want to put 30 people in a bedroom, you have to start chopping heads, okay? So you can still use them as prediction machines, but do not take them seriously for inference, because the prior will butcher the scientific process, like Kempthorne said in 1972. So these strongs that, at least from a predictive perspective, the M by M methods, such as the kernel methods, are better. Other gentlemen and ladies, and some that are neither gentlemen nor ladies, they postulate this idea of the systems approach, or system biology, system this, system that. This is not a new idea. I mean, the Pentagon has been using systems analysis in the 40s. The Afghanistan war was playing with system standards where you see the outcome, okay? And pigs do not fly. We do not have animal experiments in higher animal species that have enough replication, enough sample size. So we have to learn from flies. It's like learning about evolution from the tooth of a wild boar that was found 7,000 years ago, okay? It's a huge extrapolation. So, in conclusion, this is too much. Point one, inference and prediction are different matters, okay? Sometimes you can know the state of nature, but if the model that rules the state of nature is very complicated, you may predict very poorly because the model is too greedy. Secondly, this is the corollary, that differences in prediction ability do not necessarily indicate things about difference in the state of nature. It's just telling you that you have a robust, well-behaved prediction machine relative to another. The kernel methods are attractive not only for prediction, but also for more properly conducting GWAS, something I will argue in this little workshop that we have in the afternoon. And of course, you are warmly invited unless you are doing GWAS. Many GWAS will be GWAShed away. I think the advent of genomic data, we are now asking questions. We see that it really poses a challenge on parametric methods and to their ability to extract meaning from the evidence. And then in the question, are we going to see a future? Are my colleagues in animal breeding abandon this idea of the additive genetic model? Everybody knows it does not exist. Should we switch to semi-parametric or machine learning type techniques? Well, I don't know. I think we have to look at the fields that have succeeded in working with complexity. And none of these fields, such as artificial intelligence, robotics, geophysics, etc., have really relied too much in pet theories about the state of nature. They just have observed a system, they observe it with enough care, they measure it properly, and then they learn how the system behaves without necessarily understanding it. I'm not saying that we should abandon to try to understand matters, but from a prediction perspective, it's totally unnecessary to understand how things function. It's just a matter of evaluating different inputs, different prediction machines, and that's the end of the story. And if your objectives are narrow, like predicting whether you're going to die in 10 years, that's fair enough. I don't want to know when I'm going to die. All I know to want is I'm going to die in 10 years. If you want to make happier pigs, okay, you put your objective and I'll construct a prediction machine that hopefully on average will make better and happier pigs. Okay, so Carl, I'm done, I guess. Yes?